These methods integrate next-generation sequencing and flow cytometry imaging analysis. It can help answer interesting questions like whether some phenotypic alterations are implicated in cellular transcriptome, especially in the pollutant exposure environment. The main advantage of these simple methods for our study is that we can use it to measure the co-localization of biologically important protein to interpret the outcomes of key differential gene expression at a functional level.
The implications of this technique extend toward the diagnosis of pathological and toxicological outcomes, as a novel imaging approach facilitates the ability to connect gene expression to protein function.
Generally, individuals new to this method will struggle because of the high technical demands of the imaging and transcriptomic analysis.
To sequence the human monocyte-derived dendritic cell library, first load the sequencing and indexing reagents onto the sequencing by synthesis and indexing reagent racks, respectively, and place the racks in a laboratory-grade water bath for 1 hour until all of the ice has melted and the reagents are mixed thoroughly. While the reagents are thawing, power on the sequencer, connecting the computer to a network drive when the Do Not Eject drive appears, and launch the sequencer control software. Next, add about 100 milliliters of maintenance wash solution to each of the bottles in the sequencing by synthesis reagent rack and screw a